Hello, my dear students. Welcome to my today's online class on practical topic. You have to take up one practical paper. In that, in today's class, I will be teaching you one small unit pertaining to parasitology, paper 1. That belongs to unit 11 of practicals paper 1, and it goes like this: life history stages of Fasciola hepatica. You have studied this in theory also, I suppose. In this practical class, you have to learn about 3 larval stages that you come across while studying the life history of Fasciola hepatica, commonly called liver fluke. In Kannada, liver fluke or fasciola is called karalu hulu, okay? And this is a parasite on sheep, and it causes a disease called liver rot in sheep, okay, causing abnormalities of liver. Now, in this practical class, we shall learn about these 3 larvae. Now, let me introduce myself. I am Dr. MEERA B K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore. You can see my mobile number and mail ID on this slide, and after the class, for any queries, you are most welcome to contact me. Okay, now we shall start our class. Okay, so in this slide, I have put here few larval stages that you have to study according to the syllabus. One is MEERA larva, and the other one is Radial Larva, and the third one is Circaria Larva. All the 3 happen to be the larval stages of Fasciola hepatica. Okay. Now let's begin our class with this background. Now, for your understanding, let me first tell you about the life cycle of Fasciola hepatica in brief. You can see the schematic diagram here depicting the life cycle, and it is quite complex. It is not that easy to study, and actually it is completed in 2 different hosts. Please note, this particular parasite has 2 different hosts. As you see from the diagram, one is the sheep, that is the primary host, and the other one is the snail, that is intermediate host. Since you find 2 different hosts for the completion of the life cycle, you call Fasciola hepatica as a digenic parasite. Hence, it has been put under the order Digenia, okay, as against Monogenia, where you find only 1 host. So, this is a digenic parasite, and it completes its life cycle in 2 different hosts. One is sheep and the other one is snail. Next, the primary host is the sheep, in which the adult liver flukes live. The specific name says hepatica. Hepas means something concerned with liver. So, the parasite lives in the liver of sheep, hence the specific name hepatica, and causes a disease called liver rot, as I have already told you. Okay. Then, the intermediate host or the secondary host is a snail, that is of the genus Limnaea. There are other snails also which serve as secondary host. I will let you know little later. So the intermediate host or secondary host is the limnaea normally, in which all the larval stages what I mentioned along with another one what you are not studying, that is porocyst larva, are developed. Okay. So the adult lives in sheep and the larval stages develop in the intermediate host. Please note this. So now I am presenting before you few larval stages that you have to study as per your syllabus. With this background, first we shall study the first larval stage, that is MEERA larva. Okay. So for your convenience, again I have put here the life cycle of Fasciola hepatica in a smaller version, and below that I have put the structure of the first larva what you are going to study, that is MEERA larva. Okay. Now, first let us understand the external structure of MEERA larva, and then we will understand the internal structure. See, actually the adult liver fluke produces the eggs and the eggs are fertilized, and the fertilized eggs will release the first larval stage, that is nothing but MEERA larva. So it comes out from the fertilized egg and it leads a free life. Please note, the larva is free living whereas the adult is parasite. It is microscopic in the sense you can see it only under a microscope. It is dorsoventrally flattened, you can see from the diagram below, and you can also make out the shape, it is conical in shape. It is small, oval or conical and richly ciliated. You can see the cilia being present all over the surface of the body of the larva. Okay. Then the anterior end, produces a mobile non-ciliated, only that region is not ciliated, and that region is known as apical papilla. Okay, and in the anterior you find a mobile non-ciliated apical papilla, characteristic feature of MEERA larva. Then this larva is multicellular, right? The body is covered with ciliated epidermal plates, very important, and you can count the plates. I will show you in the next slide. There are total of 21 epidermal plates. It is fixed, 21 epidermal plates, and please note these plates are arranged in 5 rows or tiers. I will let you know a little later. And the number of plates in each tier is fixed. Don't get confused. The body is covered with ciliated epidermal plates, and the number of such plates is 21 in number, and they are arranged in 5 rows or tiers, and the number of plates in each tier is fixed. Please remember all this. We shall go to the next slide. Yeah, this tells you the magnified version of MEERA larva, showing you the external structure. You can see the apical papilla. Now let us study about the epidermal plates in each tier. See, tier 1, the uppermost one, it consists of 2 plates, you can count, 2 dorsal plates, 2 lateral plates and 2 ventral plates, totally 6. Next come to the second tier. Again there are 6 plates, 3 dorsal and 3 ventral. Then third tier, you can count, there are 3 plates, 1 dorsal and 2 ventrolateral. Tier 4 consists of 4 plates, 2 right and 2 left, and tier 5 consists of only 2 plates, 1 left and 1 right. Students, don't get confused with the number of tiers and the number of epidermal plates in each tier and the total number of epidermal plates. 21 epidermal plates in 5 tiers. Tier 1 6 plates, tier 2 6 plates, 3 plates, 4 plates, tier 5 2 plates, and I have told you the position also. In the practical class you can see the slide, you can count the number of rows and also count the number of cell plates in each row. Okay, don't get confused. Then again, continuation of other external characters. Under these epidermal plates you find a fine layer of subepidermal musculature. Sub means below, epidermal musculature. Let us see what all you find. As the name itself says, musculature means it consists of muscles. The musculature consists of outer circular muscles and inner longitudinal muscles. Under this musculature you find another layer called subepithelium. So first one is the epidermal plates. Below that the subepidermal musculature. Below that subepithelium. Okay. Now the epidermal plates, subepidermal musculature and subepithelium, all the three put together form the body wall of Meeracidium. Okay. So body wall consists of the epidermal plates, 21 in number, arranged in 5 rows, below which you find subepidermal musculature and below which you find subepithelium. All the three put together form what is known as body wall of Meeracidium. Please note, right now we are studying the external feature of first Meeracidium larva, which is the first larval stage in the life history of liver fluke. Okay. Then, now after having studied the external features, you should learn internal features also, because you can see them in the slide. Then, inside the body of Meeracidium, what all you find, you can see in the diagram. You find numerous glands, number 1, nervous tissue, number 2, protonephridia or flame cells, number 3, and germ cells. So these are the 4 major structures that you find: numerous glands, nervous tissue, protonephridia or flame cells, and germ cells. Alright. Then you find a sac-like multinucleated mass of glandular protoplasm attached to the center of apical papilla. You can see in the diagram, right below the apical papilla you find a sac-like structure. It is a multinucleated mass. It consists of granular protoplasm. Okay. Then you find a large brain with several associated nerve fibers, okay, which lie dorsally below the epidermal cell of the second tier. Okay, don't get confused with the position. So when you are talking about internal structure, you should tell what all you find. Then one by one you have to elaborate. Alright. Then continuation of this. Above the brain you find an X-shaped larval eye. You can find an X-shaped larval eye with 2 concentric pigmented cells. You can make out there labeled as eye spots, right? So above the brain you find an X-shaped larval eye, photoreceptor structures, okay, with 2 crescentic shaped pigmented cells which are called eye spots, right? Then you can see 2 concavities of these eyes, okay, and they face each other. The concavities face each other. Then they contain a refractile material that serves as lens, okay. The concavities contain the lens, okay, which is a refractile material. That is actually the photoreceptor structure. Then if you concentrate on that green colored region in the slide, they are the protonephridia or flame cells. You find a pair of them. A pair of long tubular protonephridia or flame cells are present, and you can also make out that they open to the exterior through excretory pores. Okay, through excretory pores. Then the germ cells, those almost saffron colored here in the diagram, the germ cells lie in groups, and they are referred to as germ balls. They are referred to as germ balls, which are present in the rear part, that is more towards the posterior part of the body of the Meerasidium larva. Okay. So students, when you are expected to write about Meerasidium larva, you should draw a neat label diagram, identify it properly. You have to talk about the external features, and then you have to highlight the internal features also, right? So each spotter carries about 4 marks. You have to be very careful, right? And then this slide is the one that gives a connection between the Meerasidium and larva and next larva. So I have put it here as a supplementary reading because this Meerasidium larva, can succeed, can further develop only if it comes in contact with a specific intermediate host. I have already told you, the host is the snail. Normally it is Limnaea truncatula. Limnaea truncatula. Sometimes you may see Bullinin or Planorbis species of snails also serving as intermediate hosts. But normally it is Limnaea, that is Limnaea truncatula. And the larvae which do not come across this suitable host automatically will die within 24 hours. Okay. And if they happen to come across the suitable snail host, okay, they further develop into next stage of the larva, that is sporocyst larva. Unfortunately, you are not studying the details of sporocyst larva in practicals. You may study in theory because it is not included in the syllabus. And this sporocyst larva further develops into next larva, that is Radial Larva. Okay. So this is for your supplementary reading. It do not contribute to the answer that you write in your exam. Okay. Then let us study about Radial Larva. So the Radial Larva looks like this. Let's see. The germ balls, what I was talking to you about them while talking about Meerasidium larva, the germ balls present in the the germ balls which are present in the sporocyst larva develop into Radial larva, not Meerasidia. Please note, Meerasidia larva develop into sporocyst larva, and in sporocyst larva also you find the germ balls, and these germ balls develop into Radial larva, what you are seeing in the slide. These radial larvae, larva singular, larvae plural, they come out of the sporocyst by rupturing the body wall of the sporocyst, right? They are released, and you can make out from the diagram that they are elongated and cylindrical structures. They are elongated and cylindrical structures. They have mouth at the anterior end, you can see, and you find a ring of collar cells. You find a ring of collar cells along with the birth pore. All that is labeled in the diagram, what is there on the slide. Okay. So these are all the structures present in the anterior end. You find a mouth, then a ring of collar cells along with the birth pore. Okay. We'll continue with this. So then the body wall of this larva consists of a cuticle, circular, longitudinal muscles and subepithelium. Okay. So, circular and longitudinal musculature you saw in Radial Larva also, subepithelium also you saw there. The extra thing here is cuticle. It is a parasitic adaptation. So, the body wall of Radia consists of cuticle, then circular and longitudinal muscles and subepithelium. Then you can see from the diagram, the mouth leads into pharynx, which further leads to intestine, and the intestine is lined by single layer of cells. You can make out. Sorry for the typographical error. It is not letter, it is layer. The intestine is lined by single layer of cells. Then you find numerous pharyngeal glands which open into the pharynx. Then protonephridia or the phlegm cells are highly branched, and all these phlegm cells open out through a common excretory pore. You can see the diagram. Then the body of this larva is packed with germ balls and mesenchyme cells. What you see here towards the posterior aspect, okay, they are the germ balls, right? So, the body of the larva is packed with the germ balls and mesenchyme cells. Radial larva moves and feed through the host tissue, okay? It feeds through the host tissue for its energy requirements. Then again here, to establish the connection between Radial larva and Circaria larva, I have given here supplementary reading again, which is not part of your answer, but still you should know, right? So, the larva moves inside the host, and the movement is brought about by muscular contractions assisted by collar and lappets. Okay, you must have seen while describing the structure. And normally they prefer to get into the digestive glands like liver. And now you should understand, during summer, if sufficient nourishment is available, the germ balls present in this larva give rise to second generation of radial larvae. Okay, they don't develop into next larval stage, that is okay, they don't develop into next stage, okay, but they again give rise to second generation of Radial larva. Then on the other hand, during winter season, the germ balls of radii of second generation develop into the next larval stage, that is Circaria. Please note, let me repeat, the fate of the germ balls in this larva depends on the season. During summer, sufficient nourishment if it is available, germ balls do not develop into next larval stage, that is Circaria larva. On the other hand, they develop into second daughter generation of Radial larvae. On the other hand, if the development is happening in winter season, these germ balls of radii of second generation develop into next larval stage, that is Circaria larva. Okay, this slide is a sort of supplementary reading for you. For more details, you can always refer to your theory notes or study material. Now let us study the third larval stage, that is the Circaria larva. So each of the radial larva of the second generation gives rise to around 20 Circaria larvae. Okay, so 20 Circaria larvae will come out from single Radial larva. Then, so they leave the body of the radia from the birth pore present in the anterior end. I have shown you that. and then they get into the digestive glands of the snail. And you can make out here the larva has two suckers. One sucker in the anterior end, that is called oral sucker, surrounding the mouth, and another sucker called ventral sucker or the acetabulum in the middle of the body. So in the Circaria larva you find two suckers, oral sucker and the ventral sucker. Ventral sucker is called acetabulum. Same term is retained while describing the structure of adult also. Okay. Then now Circaria larva is oval in shape with a long simple tail. You can make out here. The tail helps in swimming movements. Okay. The continuation, let's continue this. So the body layers are much similar to the previous two larval forms. Okay. Then but one difference is cuticle has backwardly directed spines. Backwardly directed spines. Body wall consists of numerous cystogenous gland cells. Okay. What do? They secrete cyst for the next larval stage. Okay. Cyst, gen means to produce. So their primary duty is to secrete the cyst for the next larval stage. Then you can make out mouth leads into a pharynx, followed by esophagus, and that is followed by intestine. And the speciality is, you can make out from the diagram or even from the slide, the intestine is inverted and U-shaped. It is in the form of inverted U shape. Okay. If you write U, ulta, so that is the shape of the intestine. Okay. So intestine is in the form of inverted U shape. At the lateral zones, you can make out so many flame cells which open into a pair of excretory tubules which finally unite to form an excretory vesicles, right? And then below that acetabulum you can see the mass of cells. They are nothing but groups of germ cells which represent the rudiments of adult genital system. Okay, genital system is generative system, that is reproductive system. Then this mature Circaria larva, it migrates to the pulmonary sac, and from the pulmonary sac it escapes into the surrounding water. Once it escapes into the surrounding water. Please note, once the Meracidium larva gets into the body of snail, it comes out only after 35 to 60 days as Circaria larva. Alright. So Meracidium larva enters the body of the intermediate host, that is Limnaea or other species of snail, stays there for 35 to 65 days, and what all happens there? All larval stages are developed there, right? Meracidium becoming radia, radia becoming second generation of radia, radia becoming the Circaria, then Circaria becoming metacercaria, that is encystment. And for all this to happen, it takes about 35 to 65 days, and it exits out of the body of the intermediate host as Circaria larva. And this Circaria larva swims in the water for some time and gets attached to the grass blades or other aquatic plants. And after some time, the larva loses its tail, it gets encysted and transforms itself into metacercaria larva. Okay. So please note, students, till now I have taught you the 3 different larval stages as per your syllabus. that you come across in the life history of liver fluke. Okay. For further details, if you want to know more, you can always log on to studyandscore.com, or you can refer to some manuals like Manual of Zoology, Invertebrates by Dr. P.S. Verma, S. Chand Publications, or you can just refer to the manuals, okay, which have been published by your own college, that is volume 1, paper 1, okay. So, with this, I complete this small unit pertaining to the practicals. Thank you for your patient listening. Any questions, do contact me. Till then, stay blessed.